Sí, vamos a hacer esto.
Yo estoy aquí en el primero, me voy después a ver a Flip Flow y me quedo aquí en detrás, que no veré mucho, pero...
Sí, bueno, no quieres más de lo que tienes fresco, pues ya es que hay un poco de los modos que tienen bastante calor, la historia es que sí, absolutamente híbrido. Pero llevo muchos años hacer cosas. Muchos. Medio vasco, medio mal. De Zaragoza. Sí, fui a hacer un máster, que entonces no era máster, para genética, digamos. Y más de mis, pero no sé pensar a la flor. No, no se jubiló. ¿Está con Juan? Que no sé qué va a jubilar María. ¿Qué dice? Antes que pasar por eso me jubilé. Sí, bueno, no sé hasta qué punto, sí, me decía mucho que esto considera mucho y decía que al final sí, se le ve muy, no sigo mucho eso, pero sí se le ve muy involucrado en eso. También, bueno, pues tiene gente del grupo que son investigadores. Pues medianamente grande para nosotros. Sí, los nombres establecidos. O sea, el de Mike, no sé si le habría aconsejado. Pues debí dejar ese hace poco a José Miras. Le conozco, le he visto presentar en dos ocasiones y me gusta, pero no se en persona con él, nada nunca. Pero sí, me gusta. Él antes estaba en el CITA. Ahora no nos vemos con los del CITA. O sea, yo veo con los del CITA como me veo contigo. Mucho, sí, sí, eso. Partimos el comedor, es un poco más de interacción. El comedor antes, ¿dónde estaba? En la residencia, que era la residencia. Y ahí había un comedor donde íbamos a comer todos. El CITA, el Pirenaico, nosotros, el IAMS, todos. Y allí, pues, quieras que no tenías un y ahí se ha quedado. Entonces, el cita es que vamos.
So, are we supposed to start?
I don't know.
¿Dónde están empezando?
Yes. Should we start? Yeah. I mean, you're in charge of this? Supporting. But I'm the first one. Liver Grape is the first project. So,
yeah.
We need to wait for the audience.
Yeah.
They're coming.
so this is going to be wine against beer. I see.
And you have to promise wine. So if you come to my session, I will bring wine.
I didn't take it in the plane.
Yeah. I promise you, at dinner you get wine. Okay. So the quicker we start, the quicker we go to dinner. So, yeah. So I would like to welcome you all to the second session. So my name is Marijke Henning. I work for Global Founders Research Institute for Agriculture, Fisheries and Foods. And I will moderate this session together with my co-moderator, Alvin from Wageningen University Research. So each presenter will have 10 minutes. And after that, there is some time for question and answers. But we are really strict to time, so we have to really stay into the timing. So online participants can ask questions in the chat or raise their hand if they want to speak. So now let me introduce the projects that will be represented today from the Apache JPI and the SysCrop joint call. So we have Diver Grape, we have White Health or Security, I'm doubting at this moment.
White Security, correct.
Okay. Wineland Dip and Rico Bar. So now let's start with the first one, Diver Grape, and I want to introduce Diego. Interligo. I'm not sure how to pronounce your name.
Diego Maradona.
Yeah.
I wish I was a football player and not a scientist.
I will start your presentation and then hand over to you. So you have 10 minutes.
Yes. So thank you. This project, thanks for coming to this session. It's about the effects of genetic diversity, the natural diversity on some ecophysiological responses of grapevines or water use. And I'm working at the Certification Research Center, which is a joint research center among different institutions in Spain, CSIC, the University of Valencia, and the regional government. Well, as you know, wine grapes are very important crops. It's a very important crop all over Europe, mainly in the Mediterranean area, but also in Central Europe. And with climate change, I know that wine grapes are grown even in England and other places. And there is a very large diversity in the genetic background with many rootstocks, varieties. But unfortunately, very little of this diversity is known, and particularly for traits such as water use and aspects related with water, because most of the grapevine cultivation is based on a few varieties, despite this large diversity. So our scientific objective indeed was to understand the magnitude of this diversity and the traits, the ecophysiological traits responsible offered by the rootstock, the cultivars, and the clones. So the perennial crop grown mainly with a root. The rootstock is a genetic material, and the seed is a different genetic material grafted. And of course, there are interactions between the two parts of the plant. And basically, the rootstock is affecting the rooting depth, the hydraulic conductance, the vigor, and the xylem architecture, while the CO part is affecting the stomata, the leaf area, and other characteristics. So we want to gain insights into the diversity, the response brought by these parts of the genetic material. We are a very small project, and I take advantage actually to say that sometimes I think small projects, when we have a specific objective, are much better to manage and to work than very large projects that many times are stimulated by the European Union, ERANETS. Here we are just 3 partners, so everything is much easier. And I think the
coordination has been facilitated. We work in 3 quite different environments, from Valencia in Spain with very low precipitation rate and high temperature, all the way to Bordeaux, where the rainfall is much higher, and probably Austria, which has the rainfall is not that high, but the growing degree days is definitely smaller. So we work in a diversity of environments, and this is the opportunity brought by this type of project that we can basically test our scientific hypothesis in different environments. Well, this is to say that actually when we first submitted the proposal, we also had an Italian partner that unfortunately could not be funded. And so we had to remove that part, which was kind of sad, but also I want to explain here that basically they couldn't be funded. And we are working on some different type of experimental platforms where we innovation the same genetic material, either the seed or the rootstock, but in different environments. With environment, I mean in different soil conditions, also different practical agronomic practices, and so to have a diverse type of responses. These are the different terroirs in viticultural terms. This is the Spanish vineyard, which is a global type of system. The Austrian vineyard, which is a vertical shoot positioning. The French vineyard, which is also a vertical shoot positioning. So there are many differences in the 3 sites, and not only the environment itself, but also the agronomic practices are different, and this will also, of course, play a role in the responses we are looking at. Well, just some results. We started last year, so we are really working on this. We also have to the results we have obtained, we have to still analyze them, but I will show you some results. For instance, this is the work done in France by IBRA in Kyoto, where basically they are looking at stomatal conductance in 3 different varieties and in different rootstocks, and they want to see basically if there are some trends among the different genetic material. They found that there is the season is making a big role, so there are large variations along the season, but there are also some trends. For instance, the lowest values in stomatal conductance are found in Grenache, while the different rootstocks, they don't seem to play a very important role, and they found a lower effect than what we would initially think in this case in France. Also, we are doing some similar experiment in Spain, where we work in the same genetic material, and we have well, results from 2 seasons, 2023 and 2024, that we are still analyzing. So these are very preliminary results that I could analyze coming year. Well, we know that stomatal conductance is very much affected by the plant water status indeed. So we basically are related stomatal conductance with the stem water potential, the midday stem water potential, because regardless of the genetic material, indeed the water status of the vine is affecting the stomatal conductance. So we are analyzing these relationships. We will look into the residuals, but so far, for instance, we are finding that the one rootstock, in this case SO4, has lower stomatal conductance for a given plant water status in 2 seasons, maybe more clearly in 1 season than in the other, but we are going to do all the statistics with this, but we are finding this. And interestingly enough, in Austria, which is a very different environment, as you've seen, also very different type of training system, but they have found when they look at the seasonal variation of stomatal conductance in these rootstocks, they find that also rootstock SO4 has a lower stomatal conductance. What does this mean in terms of the vine performance or of the water use is something that we are going to analyze over the course of the project, because stomatal conductance is a measurement done at the leaf level, and then we need to analyze also the wall canopy area, the light interception, in order to upscale the values of stomatal conductance to the whole vine. And so for this we will be applying models in order to obtain the water use, but we are still working on that, and I couldn't present with this type of results. Anyway, that's just to give you some hints of what we are doing. Also, we are looking at the nutrient status of the vines to see if there are differences. So analyzing, for example, facial nitrogen across the varieties and across the rootstocks. Also looking at the mast nitrogen in the mast, as an indicator of the nitrogen capacity of the capacity of the vine to obtain nitrogen, and we are finding some differences among the variety in one case, among the rootstock, and now the work is to make sense out of all this data we are obtaining. And also, indeed, I mean, we are agronomists, most of us, or I'm an agronomist, so I'm very interested also in the yield, the wall vine water use efficiency, so we are analyzing the yield. But it is to say that the yield in grapevine as a perennial crop is a trait which is difficult to interpret in terms that is influenced by many things, also particularly by fruit set, for instance. There's also some differences in the environmental conditions around flowering and fruit set might affect fruit set indeed, and then affect yield, and that's why we have still to work on this. But for instance, we see some responses among the varieties, also depending on the rootstock. In fact, there could be interactions between the rootstock and the variety, and some varieties they might work well with one rootstock, some others no, and this is the type of data indeed that we are obtaining. Well, the next steps for the future is, of course, finishing to analyze the data we have just obtained this season, obtain a third season of results. Working with perennial crops is fundamental to work at least during 3 seasons. Integrate all the data we are obtaining about stomatal conductance and plant water relation across sites and season in order to do an integrative type of analysis. We are targeting to write common publications in high impact factor journals if possible. So we would like to have a few publications, but in good journals. And indeed, also it is important, of course, to connect with the stakeholders. So we are planning to publish in the ICE technical reviews in different languages. This is a journal for growers, also to do field days with local growers to show them the results, and also to connect with the origin denomination in the different regions, which are basically the Growers Association, where we will like to provide them with the results we are obtaining, of course, using a different language than the one I use here for a successful technology transfer. So that's all from my side, and just to say that this is the research team with Professor Gregory Gambetta, very well-known ecophysiologist from INCAE, Jose Carlos Herrera from Bogu, and my colleague Ignacio Huesa, which is a young scientist, and myself that we work at FIDE, the Seed Research Center. Thank you.
So thank you for your presentation, perfectly in time. So we still have a few minutes time for questions. So are there questions coming from the audience?
Thank you for the presentation. Yeah, I'm from Stellenbosch in Cape Town, so grapevine is very close to my heart. Can you, so I'm not a grapevine agronomist. Can you explain the relation between the stomata conductance, water usage, water availability, and maybe temperature? How does those things fluctuate? What's the relationship between them?
Yes, stomatal conductance indeed is a variable related to the degree of opening of the stomata.
Yeah.
So the higher stomatal conductance, the water use, the higher the water use, and also the higher the photosynthesis. Because it is how open are the stomata. However, then the wall vine water use, it is related to the stomatal conductance, but also the leaf area. So you might have high stomatal conductance, but then if you have low leaf area or low light intersection, the water use is affected, and also by the environment. So the vapor pressure deficit, which is related to temperature, is the driver for water use. So if you have a day like today, even if you have the stomata very open, because it's very humid and overcast, the water use is low. While if you in a hot and warm day for a same stomatal conductance, you have a lot of water use because you have a lot of demand from the environment. So it is just an indicator of the water use, but it's not the water use, which is a parameter much more complex related to many other environmental and plant factors.
Does this change during plant growth? Say you have your normal phase, then you have fruit set, and obviously then you have berry development. Does that change?
Sure, it changes because it changes due to the anatomy of the leaf, leaf ontogeny, it changes. Also because tomatal conductance is also related to endogenous factor, and also not just the water part, also the zinc salt part. So the carbon demand by the crop might change during the season, and this has affects feedback or feed forward on tomatal conductance. So there are seasonal trends, and that's indeed independently of the environment, also because of the physiology of the plant changes. And so there could be the difference. Also, depending on the leaf type, in a grapevine, if you take an old leaf or a young leaf, their tomatal conductance will be different. So it is a challenge actually to integrate a single value you obtain in one leaf to the entire canopy level.
What is the rationale about the choice of the rootstock? Because you choose 3 rootstock, different rootstock. How did you choose this rootstock? Or I just on the other question is, do you find some interaction between rootstock on the varieties of grapevine?
So for the first question, the nice answer would be we choose the different rootstocks based on basically different behavior they could have. The reality is that we had to start with an existing vineyards. So we basically we looked for vineyards that had the same rootstocks in the different places, and we took advantage of what was already available, because the project is for 3 years. If we start a plantation now, then we will be able to work on this in the next project, and we have to work on this. So basically we took advantage of existing vineyards with different rootstocks. But yes, for instance, 103%, it is a very vigorous rootstock. SO4 could be a less vigorous rootstock. So those rootstocks were actually chosen because they're different. And yet there are many possible interactions, because there is in the grafting union, where you graft the seal into the rootstock, there could be incompatibilities between the variety and the rootstock. And those incompatibilities affect a lot on the hydraulic conductance, particularly at the neck, at the union. And those will affect indeed the world vine performance. So it is already well documented that some varieties might work well with some rootstocks and not with others, because of this different level of compatibility in the grafting. So.
One small question because of time.
So I'm just curious about whether you are considering investigating the microbiome at all, or interactions with microorganisms in the environment.
Well, we are not. I think we are busy enough with, or we already have. Definitely, that will be a next step, and this actually takes advantage to. So all this, the environment is playing a role. No, the rootstock, depending on the soil, will perform in different way. If you have a dead soil, a shallow soil, and probably also maybe the possibility of looking at the microbiome. We are not that level yet. I know some colleagues in Logroño, they are working on that, for instance, looking at the microbiome depending on the rootstock, if there is the difference. This is still more fundamental type of approach. Yeah. Next project, maybe.
Thank you very much. So we go on to the next, the bit security.
So my name is Fiona Dutton, and I'm from University College Dublin in Ireland, and I'm the coordinator of the Big Security Project. And the idea behind this, as I said before, was, well, first, it's an important crop. As most of you know, it's responsible for 20% of human calories in going up all the time. And we work in this project with the 2 types of wheat. We work with red wheat and we work with durum wheat. I'll go through the partners in a minute. So, as I said earlier on in the pitch, You know, the main focus for wheat breeding for a century was yield, with, of course, looking at disease resistance and factors like that. But the main selection factor was yield under high input production conditions, high input pesticides and high input fertilizer conditions. So, you know, we all know the challenges that are out there now in terms of climate change, in terms of the Green Deal, and also in terms of nutrition. I'm a pathologist, I'm a plant pathologist. I've worked on wheat for 30 years. And very naively, about 15 years ago, I went to one of the big breeders and I said, I'm really interested in the relationship between Fusarium head blight disease and nutrition. And they said, we're interested in the Fusarium, we don't care about the nutrition. But that's changed, the world's changed now. And so really this project is looking at all the things in a more integrated fashion. So it's called wheat security. It's looking at how we manage them in terms of inputs. So we're looking at low versus normal nitrogen levels. We're looking at plus and minus biostimulants. We're looking at the impact of the environment and the genotype, of course, being very important, on stress resilience, abiotic stress resilience, particularly direct experiments in plants, and biotic stress resilience across multi-locational sites. We're looking at the impact of variety on the microbiome and the soil microbiome specifically. We're also allied with other grants where we're looking at the impact on the head microbiome of wheat as well, and the foliar microbiome. And in Spain, Carlos is looking at grain quality, so the quality parameters of the grain. And Tancredi and UCD, who's also involved in another project, is taking that all together in terms of network and data analysis to see, are there specific are there impacts of the variety on the microbiome and on soil health? Are there particular microbial associations associated with particular genotypes? And how do genotypes perform across different environments in a holistic manner? So the team, there's a few of us in UCD, there's also people in Italy, in CREA and in Unibo, there are people in INRAE in France, there are people in Chagos in Ireland, and there are people in University of Copenhagen, and University of Bologna and University of Cordoba and University of Barcelona, and then several industry partners from SMEs to larger partners, and they all bring different skills to the project. So where are we? We're just at the end of the first year of field trial, so I don't have huge results to show you. But just to overview the project, we have the work package 1 where we have the data development integration. We really spent quite a bit of time, Sophie and I really spent a lot of time on this because there's not a lot of data available already for wheat. So rather than reinvent the wheel, we look at what's out there already. And then the big thing in the middle is really looking at the field trials. So we have multi-locational field trials in 2 different locations, generally per country, with the exception of Denmark. But in Denmark, Italy, Ireland, France, and Spain, where we're growing a common set of wheat varieties and region-specific varieties. We're growing both bread wheat and durum wheat, limited durum wheat in Ireland. So we thought it wouldn't do well, it actually is doing surprisingly well. So we will replace Italian wheat, durum wheat. So we're doing those trials. We also have large populations for GWAS analysis for specific traits, which I won't go into too much today, but we have those deployed in Italy, in Ireland, and in France. We've taken in other partners. John is looking at some of the nested association mapping population. So in the multi-locational trials, we're looking at the microbiome, looking at the soil characteristics, the environment. We're looking obviously at the agronomy of the crop and how it's performing. And we take those back. There's a lot of stakeholder engagement. Stakeholders come and visit the farms and the varieties. This is very important in some countries, for example in Ireland, where the presumption is that you cannot grow bread wheat. It's quite surprising for the farmers to see that you can, and also to see that there's lots they can do in terms of sustainability. So I won't go through this in great detail, just to mention that we have the database to inform us. We're doing the field trials. We have agronomy, we have phenomic analysis, genomic analysis, and modelling, and machine learning, bringing all the data together at the end. We were a bit caught in the timing of the project. We're now at the end of year 1 field trial. We're doing year 2 field trial. We're meant to do 2 years field trials in the sites, and that's what we're doing. And the key findings to date, well, we did a lot of parsing of the background data to select the common varieties based on genetic diversity, disease resistance, known yield traits, etc. And Sophie did a lot of work on that, and we came up with a list. We selected varieties that we presume would go also well in one variety and not in another, and that's really important. I'll come back to in a minute. We just completed year 1 trials. This is the location of the trials. So in Ireland, we had 2 trials, one in Dublin on the UCD farm and my university farm, one in Carlo. We had trials in Spain, and another one next year in Barcelona, and Venezuela, Ravina, and then in Copenhagen as well this year. So 1 or 2 of the trials last year, it was such a bad year, we couldn't get them into the ground, for example, in Copenhagen. So, and just to show you here, for example, they're not all trials are the same, but there are extra things in different countries. For example, we're using the phenotyping platform for drought in INRI to look at the drought tolerance of some of this material. So that's one outcome so far, early days, but we see that there is a lot of variation in response to nitrogen, and a lot of site-specific variation. In the glasshouse, we're also doing experiments to augment the field stuff. So, for example, what you're looking at here is the plant array phenotype in Crea, in Venezuela, with Elizabetha and Delfina. And they've been using this platform to screen our genotypes to look at their drought tolerance. And for those who don't know the plant array, you're essentially a very fine balance. You're weighing the plant, it's the exact amount of water on our drought period, and you're looking at the impact on biomass. And they use that to screen the response of different varieties to drought, and also the different varieties plus and minus the seed biostimulant treatment. And this just, each one of these graphs represents a different variety, the weight on the y-axis, and on the x-axis here you have different time points. So they're weighed across a continuum of time. And there's a very obvious difference in the weight of the cultivars, i.e. the response to the drought. Here you have the drought period, you can almost point it out yourself here. It starts dipping, where the weight starts falling down. Plants were droughted. And just to zone in one of those, just to look at a biostimulant effect. So here, Delfina and Elizabetha put a seed treatment biostimulant on the plants. We had the same biostimulant and a few others out tested in the field. And what you see here is minus the biostimulant. So this is the weight of the plant over time. Here's where you have the inception of the drought period, the kickoff of the drought period. You see the weight starting to plummet. You'll see there's a less of a plummet with the biostimulant. And then they re-watered again. And now they're doing much more finessed experiments, zoning in here, well, if it's not just re-wetted completely again, is that effect longer? But actually it was quite surprising. I didn't expect such an effect. And then just the last thing, and I don't have any great results on this yet, but it's anecdotal from the field trial managers, but I know it's true in Ireland. We didn't get what we expected. We planted varieties in Ireland that we selected based on what's grown in Ireland, a number of varieties, and then Italian varieties, Spanish varieties, etc. The varieties that we would have hypothesized to perform well performed okay. Some of the Italian ones did amazingly in Ireland, and a really bad wet year. So it also tells you that the agrobiodiversity, that we haven't really exploited it fully for all the different agroclimatic regions. And we're even contemplating this year, if you can see my cursor there, putting some trials up in one of the most extreme environments in Ireland, which is in the northwest. So the next step for us is to complete the field trials and the glasshouse trials. We have biostimulants that worked. I know at least one of the biostimulants in Ireland was having an effect of an increase in yield of 18%, which is a seed treatment combined with a foliar, which was great. And we're optimizing and assessing other parameters. We're doing the microbiome analysis at the moment, and UCD is responsible for the microbiome analysis. We do a lot of work on the relationship between plant genotype, wheat particularly, also barley and oats, plant genotype, microbiome, and diseases, foliar diseases and fusarium head blight disease. We're doing that at the moment there. And Thomas in University of Copenhagen is looking at the leaf physiology. So he has a sort of biochemical test to predict yield based on leaf physiology, enzyme, multi-enzyme tests. And Tancredi will bring all the data together, hopefully, in the end, to tell us what's happening. So he'll do the data modeling and the association studies, And really just testing the hypothesis that deploying more diverse varieties will pay off, all better support lower inputs, more sustainable agricultural systems. Of course, that's a very isolated island of wheat and so on, but it's a start. But the idea that the impact will be to inform breeding. And I mean, for us, it's brilliant if we can demonstrate to farmers and to breeders that we are in the stakeholder events, that there's an added value potentially to a particular variety, perhaps in terms of disease. We have really high disease levels in Ireland. We have a very humid climate. And so we're seeing big effects already from bringing some in some of that germplasm. And basically we want to validate some of those tools and resources to breeders and to farmers and to the end users. So if you want to contact me, that's my email. I'm based in UCD in Dublin. I'm very happy to collaborate on any cereal projects in general or intercropping, because we'd really like to look beyond this now to some intercropping and rotational projects as well, taking what we're finding. Marco in Unibo will be doing a lot of the sort of preliminary field-based analysis, so we will have preliminary results in about, I think our mission is there in December. We plan to have preliminary field trial results validate for them, or if you want to attend the stakeholder.
Okay, thank you very much, Fiona. Also here we have a few minutes time for questions. I want to know from the audience, are there questions? Nothing immediately?
Well, maybe
Yeah.
about this last point that you mentioned, intercropping. What are the standard intercropping stage for intercropping, or what are the that are used in Ireland, or what are the crops that are grown after each other in Ireland? That we saw before a rotation of some grassland between cereals. So yeah, can you please comment?
Yeah, so the problem with Ireland is it's treated as a whole and it's very diverse. So so it depends on the region. For example, you can have potatoes in the northwest, you can have root-based crops rotated, you have a lot of grass-based rotations. So a number of years in cereals and then rotated into grass. But dairy being the biggest agricultural economy in Ireland. So there's a move back always towards grass. Now we're seeing more oilseed rape coming. We didn't have so much of it in the past. And that's a really growing industry actually over the last few years. But I would say the main one is that you're rotating out into grass or into potatoes.
I have a question, really sorry, because it's not something that I'm familiar with. Apologies if it's stupid.
No.
But the drought experiment that you presented, could you also interpret that it makes no difference whether there is a stimulant or not? Because at the end of the day, they always recover at the same speed.
You come back to the same thing. Yes, but there is a little caveat in that experiment that they overcompensated at the end. There should have been a slow up, if you see what I mean. There was a slow down, and then there was a fast up. But it was just to see, is it worth doing any further? And now what they're doing is a much more refined, because the machinery is limited. And they wanted first to check after the bottom crop, is there an effect? But based on that data, you're right, you could interpret it that that's growth stage. Now it's not real at that growth stage. An effect. The one thing I would say on that is that we do know with not that biostimulant, but another one they're testing that there has been an effect on drought tolerance in the field. Now, from other studies, not in this project, but that's why we took it into this project, because somebody had demonstrated an effect of drought.
And could you kind of extrapolate this to field work in the sense that you just need to wait for until it rains a lot and then everything will go back to that?
Yeah, it rains a lot. You know, I mean, that's not reality, you know, and that I know from some of the preliminary studies that when they ramp up slowly, you get a lot more depth. You're not seeing that there, you know. So it's a good first trial just to see, is it worth pursuing?
Okay, thank you. I think there is still one question online, or Isabel, if you want, you can start.
Yeah, just a more general question. Thank you for your presentation. Actually, most of the wheat production at the European level is from intensive production. what do you think about the future for this intensive production, who will likely suffer to climate change, but which will as well keep its economic model?
Yeah, so I mean, there's two different things there. I mean, in terms of food security, we have intensive production around, we have an alternative models there to stay. I would say we're not very good in crop science in general at economic modeling in the way that they are in animal science. So Irish agriculture is dominated by animal science. And I look at the very refined bioeconomic models they have now. For example, Carl O'Donoghue at the University of Galway, which takes in the climate change impact. It's not just, it's bioeconomic, bio takes in the climate change aspect. So to go back and answer your question directly, what do I think? I think we will move away from the intensive system that we have at the moment. I personally think that, we need to go back and look at some of the more alternative crops. I see, for example, people pushing intensive wheat into areas of Ireland, which were very good for barley traditionally, very good for oats traditionally, and other things. And rather than, I think the answer is very complex, and that for the intensive areas in Europe, we can change the practices in terms of germplasm and inputs and maintain to quite a high level, not the same level, but to quite a high level, the productivity, while at the same time integrating other crops much more so in the other peripheral areas. I think that's the answer in terms of cereals to food security as well.
Okay, thank you.
Thank you. So we will move on to the next one. So I invite Stefan Nicholas from INRAE France to present my identity. I don't know. We are presenting to so. No. No, it's in German and it's
Yes.
Yeah.
I
hope.
I don't speak German. Okay, I will present Mining. That means mining allelic diversity of maize monoices from tolerance to abiotic and biotic stress. And I am the coordinator. This outline, introduction, objective, preliminary result, and stakeholder. Just a brief introduction. Maize genetic diversity was shaped by domestication, expansion, and adaptation to different environments. Maize have been domesticated in Mexico, close to the Mexican, then spread to North and South America, and then were introduced in Europe and in the world after that, after the discovery of America by Christopher Columbus. For this reason, non-vessels were well adapted to different local agroclimatic conditions and human use. And we think that the non-vessels carry original and promising allele for breeding to fast climatic change, low input agriculture, and new use. We hypothesize that each non-vessels were locally adapted to different use. And the second thing that we have in mind when we are working on a maize genetic diversity is there is a big transition from non-vessels to modern F1 inbreeds that lead to partial loss of diversity. Non-vessels following the discovery that we can combine, we can derive hybrid line for non-vessels, then combine in hybrid and reproduce clone as F1 inbreeds, lead to the first F1 inbreeds, and after there is a recurrent reciprocal selection that erode progressively the genetic diversity. And when we trace back to the first hybrid lines that have been the founder of modern maize breeding programs in the US and in Europe, you find very few non-vessels that from which hybrid lines are derived. And it's a reason for which we think that there's a lot of non-vessels that could be interesting to enlarge the genetic diversity. Unfortunately, thanks to our previous collect, large numbers of flow masses have been collected and maintained in gene banks, but also in situ by farmers in South America, in Mexico, and Africa, and so on. However, there is limited use of inbreeding program by farmers due to lack of characterization, gap in performance, and large within genetic diversity is a very big drawback to the characterization of genetic diversity of flow masses. On the question that we have in this project is how to identify promising flow masses on adaptative allele as key, because we do before. And we have this global objective that I presented in the pitch previously. In the first package, we try to evaluate what is the phenotypic response of flow masses to abiotic stress, and I will come back to how we do that. In our package 2, we try to identify genomic regions involved in adaptation by genotyping these flow masses in the enable, because there is large within diversity, and we evaluate, we identify this region by GWAS on threat, but as an environmental variable from a collection site of flow masses. On the package 4 is dedicated to evaluation of the performance of flow masses in compressed environment using genomic and phenomic predictions. In which we try to use model to predict what is the threats in flow masses based on genotype or not phenotype. In our package 4, we try to focus more on what if there is beneficial alleles for tolerance to abiotic and biotic stress that could enlarge genetic diversity by using targeting sequencing approach. On the first world package, that's led by colleagues from Italy and Spain, the question is what is the phenometric variation of flow masses in compressed environment to different abiotic spreads, and we choose nitrogen, growth, heat, cold, and corn bower. And we select 341 temperate flow masses among 1,000 flow masses that we genotyped previously, based on sediment criteria that is very useful to that optimize the prediction of other flow masses, but based on this set of flow masses. We choose to be temperate, to have geolocalization, to perform landscape genomic, and we try, but we did not arrive too much, to have restricted flowing time window to evaluate stress tolerance. Here you have the selection of flow masses. In blue is flow masses that constrain because it's very interesting, and after we let the algorithm explore the genetic space. In orange, it's flow masses that have been chosen in addition on blue flow masses that have been selected by different gene banks. On the left, you have the origin of flow masses. As you can look, it's largely spread across Europe, mostly around the Mediterranean basin, but we have some flow masses from Germany and North Europe. But we try to remove them to avoid to have too much earlier flow masses. On the first year, we do seed multiplication for feed and platform. And it's done, and now the trial are ongoing in the field. And here is a different experiment that we have. We have root phenotyping in 2 platforms. In UC Louvain with a beautiful hydroponic platform in the University of Bologna on Horizontal to explore what is the root architecture of these flow masses. And we have also the big part is the field experiment with high throughput phenotyping. And we have high throughput tools with harvest and post-harvest measurements with drone and so on in 2 locations in France. And after we have field evaluation across an European network with 6 trial locations with contrasted treatments in 2024. And we just finished harvesting of all trials. Not totally. There is just 1 that are not finished for the moment. On the platform experiments are ongoing. On next step is analysis of response of the threat to different treatments and comparisons and so on in platform. But the first preliminary result was very interesting. We observed difference of 8 for between growth and water and water deficit treatment, for example. On the second part is the multi-environmental and threat association study on landscape genomics. And we developed and set up GWAS method for non-masses using allelic frequency on environmental variable. And here is an example on 263 French non-masses that have been genotyped and for which we have phenotyping this previous experiment in field, old experiments, so recycling of data. And we find very beautiful peaks around genes that are involved in kernel one number, on color, grain color, and so on, but also on near composition. We have also peaks for GWAS, environmental GWAS, meaning that we find some association peaks that are associated with environmental variables from the collection set of non-masses. And now we perform DNA-based genotyping of 341 non-masses, and the 600K genotyping to densify the genotyping to perform GWAS are ongoing, and we try to develop a method for predicting allelic frequency. For the targeting sequencing, we have the design. On the next step is diversity analysis, GWAS on traits for real platform and environmental variables. The last one is we identify promising allelic and functional diversity using targeting sequencing on RNA-seq. We have the 50K array to compare non-masses and hybrid labs. We have a lot of non-masses that have been genotyped in this project, previous project, and so on. A lot of hybrid labs, but the problem of SNP array is there is a certain bias, and we didn't discover new alleles. And we try in this project to develop new targeting sequencing approach to discover and find new SNP on haplotype in non-masses and hybrid labs. On the very first result was very interesting. We have preliminary experiment on targeting pool sequencing. We tried some 110 genomic regions under 14 criteria involved in adaptation and tolerance to abiotic stress. And we sequence in this region using DNA for non-masses and DNA for hybrid labs. And we have a pipeline, we develop a pipeline, we have haplotype, and we observe that we have new haplotypes that are not present in the line panel that cover a large diversity base. And what is very interesting here is different haplotype in different regions. Skip on the black one is a new haplotype that can discover in different genomic regions. And here is just a picture of haplotype map. And we have clear pattern of haplotype frequency variation between Europe and America that follow environmental variation. That means we have mostly have some interesting haplotype that could be eventually transferred in hybrid line books if it's not present. And now we are designing a new targeting experiment for more sample. And last one, mostly the most interesting for characterizing gene bank, non-masses in gene banks, we develop genomic selection approach for predicting threat on genetics, for predicting adaptation to future climates. And here just an example that we recycle some data from previous publication. We use flowering time because it's a hereditary threat. We do genomic prediction on 1,000 temperature non-masses using GPLOT approach, and we have a very clear pattern of flowering time variation that corresponding to geographical origin of non-masses. And we have, I didn't show it, but we have good accuracy for predicting different threats, agronomic threats like flowering time and composition. And we are pretty certain that using a mainland project, we can predict other threats that we are not so present in the experiment that we have previous project. I have to finish. Yeah. By a genomic offset, we have preliminary results that are very interesting. You have just Italian non-masses. We measure genomic offset. More is red, more you are malad, less you are adapted to the regions. And it's very beautiful because when you are blue, you are close to the Italian non-masses. Where you are red, you are distant in terms of environments from Italian resources. That's on just stakeholders, we have good connection with gen banks, pretty good connection with breeders. We lack of connection with farmers. For scientific, it's okay. On what is missing in our partnership on stakeholder is more citizen. I will be very happy if we have colleagues that will be engaged in this kind of patrimonial valorization, fusion of land resources is very beautiful. And I think it contributes to know our patrimony. And just what support is needed to increase our part. Clearly, we need support for European genetic resources and European research infrastructure for that. It's very precious. I believe it's very important for us and for the future to have these genetic resources that have been maintained, but that are also living in the field. And we need support for that, clearly, because develop methods, but it's not possible to implement in general. And that's all the partnership. A lot of good colleagues from France, Italy, Belgium, also Turkish colleagues, and Spain colleagues. I will thank all people that collect and maintain more resources on the line collection from different Germans. It's very precious for our work in the future. Thanks a lot. Sorry for.
Thank you. Thank you. So maybe one small question or one question. There is no question. But they can talk to you in a coffee break if they have a question. So then I invite the last speaker. So welcome back by Esterno. Your last name, Katura? Also for you. All right, we are just ready to continue.
Good afternoon again. My name is Ernesto Igartua. Don't worry, it's also challenging to spell it. So you're in good company. Okay. Is there anything to just this? Okay, so why this project? Well, as I said before, barley is a staple of European agriculture. It's a highly diverse and adaptive crop. We love it. It's really one of the most adaptive crops we have. And as you know, the Green Deal mandate less nitrogen fertilization, which is probably a sensible thing to do. And also, and as mentioned by all the other projects in this panel, this crop is challenged by climate change, by many abiotic and biotic stresses. This is important because plant breeding, a very conservative discipline, used to have the target areas very well defined. Now, plant breeding is aiming at a moving target. We have to help them. We have to think 20 years ahead of what the conditions will be 20 years from now. We have this problem, how to predict the performance of future varieties. We can test them in the environments we have now, but we don't have the environments of the future exactly. And also we must have a better understanding and leverage of crop ecosystems. With this, I mean mostly soil microbiota. So this is already shown that this is a graphical summary of the project, and we construct this project on quite good knowledge. On the left-hand part of the slide, you see names of past projects that in which we studied a lot of the traditional genetic diversity that exists in Europe and in the Mediterranean area. And with that, we have constructed a core set of about 100 heritage and landrace genotypes. Heritage is another name for old, okay? So fancier. And also we have some introgression lines, which I'm not going to into detail with that. And these are, we are testing these old genotypes because they maybe they do good enough under the current conditions of more stressful climate and lower nitrogen in the soil. So let's have another look at that. It's a revisiting of the old diversity. This is done in actually 6 field trials across Europe. We have many colleagues here, like in UCD, we have Sonia Negrao and Tancredi Caruso, for instance, and in CREA, we have Alessandro Tondelli, so many, and so some of them have been mentioned already. The first field trials, we are halfway in the other projects in the project. The first field trials were harvested this year and under low nitrogen, natural low nitrogen. And here, for instance, you can see the orange lines represent Mediterranean landraces. This is in Spain, in Zaragoza, my hometown. The gray lines are heritage varieties, spring-to-row barleys from Central Europe. The blue lines are modern varieties, and the landraces are doing better than we expected, although some of these lodged completely and had to be harvested by hand. We also have results from other sites, and they don't look as promising for all varieties as this one, but this was good enough, and we are really curious to see what the second year will bring. We think that some of these varieties could actually be recommended for direct use in some environments. So that will be probably a nice deliverable from the project. Another thing we're doing is we have selected a microcore panel of just 10 genotypes, representing as much as possible the diversity found in this 100 genotype collection. And we are subjecting these genotypes to all sorts of torture with abiotic stresses, with heat, drought, high CO2, water logging, and then we have a specific section on logic. The first results have already been collected, and we have found very nice genotypic diversity for heat tolerance with heat wave in greenhouse shifting experiments. We have quite a few experiments with dealing with drought, with many of the partners in the project working with this, and also with high CO2, which is done at the University of Tartu. We also find really nice genotypic differences, and this, for instance, is stomatal density difference between the genotypes. Many things could be said about this, but it's a very interesting result. We are using the same lysimeter platform in CREA that I think Fiona presented just a few minutes ago. And we also find genotypic differences for drought tolerance. Water logging, this is done mostly at the University College Dublin by Sonia Negrão, and she's also using lots of physiological measurements with remote sensors, drones and stuff. And actually, she found really nice differences in gene components already between waterlogged and controlled treatment. So we are aiming at having this sort of table by the end of the project, in which we will identify stress-related traits, and we will define the tolerance or susceptibility, the range of variation across these 10 genotypes for these traits. As an additional activity which was not planned, we are doing nested association mapping population crossing one leaf germplasm LGGR by the other 9 lines. So we will generate material to go on and explore further this stress tolerance. I think I'm going to skip this. It's a little mining in all the genotypes, because we have whole genome sequencing or extra capture data for all of them. And I will come to this other part of the project. So we have this group of modelers at the University of Milano, Roberto Confalonieri and Livia Paleari, who are doing crop modeling. They construct crop growth models, and they use them to improve on genomic prediction. So they try to master the genotype by environment interaction, which is huge in crops, as you know. They try to find the traits behind the genotype by environment interaction and model them and see if they can predict better the performance, the gene performance of future genotypes using these parameters. This looks like science fiction. It looked to me in the beginning. They are showing quite interesting results. They have already improved this, what they call hybrid crop growth models. They have modeled many future climate scenarios, and they are already working on the model genomic prediction, which is quite good for phenology so far. Well, phenology is the easy one, but it looks quite promising. And they also aim at breeding toolkits by looking at which traits will be more favored in each environment and trying to predict which ideal types of barley varieties will be better in the future. Finally, we have the microbiome diversity, which we are exploring. This is mostly the work of Tancredi Caruso in UCD and Agata Daskowska College in University of Silesia, Katowice. And we are exploring microbiome crop diversity with two additional dimensions. One is breeding history, because it is already stated that there is genotypic specificity about which microbes are recruited in the rhizosphere. So we are testing 6 varieties, 2 land races, 2 heritage varieties, 2 modern varieties. And the second dimension is nitrogen. So we have collected the rhizosphere in low nitrogen soils and standard nitrogen, and these are being analyzed right now. But within the project, we were lucky that Grady and Sonia already had samples of microbiome in a study comparing control and waterlogged barley genotypes, and they have found quite different microbial networks comparing the two environments, and also the type of symbionts that are more abundant in waterlogging and control conditions are quite different. Stakeholders, we have this year, second year, we are going to run demonstration fields in Spain, Turkey, and Italy. We have several good partners who run networks of field trials for farmers, and we have identified genotypes who may be interesting for them. We have interest for the product material from one Spanish company, and then we have a very active communication and dissemination work package. We hired a communication agency, and it's providing us, well, it's helping us to be very active in social media and in press releases. Next steps and timeline. Well, everything is going according to plans, which is the first time I say this for a project. And thanks mostly to Ruben, my postdoc, who is actually doing the management of the entire project. So thanks, Ruben. I know you're listening to me. We will have a second and third field season, and the control condition experiments are almost done, but we are having a meeting next week to find out if we need to do more. So this is the Recopart Consortium. It is coordinated by CSIC, by myself, and then these are the other partners in Finland, Turkey, in Italy, in Poland, in Estonia, and in Ireland. And you have the social media there, so if you want to be our followers, please go ahead. What support is needed to increase your impact? Future projects. That was easy. My question to you, we are halfway in the project. We are open to receive any recommendations, particularly from very close projects like the ones that were presented today. That's all.
Thank you, Nelson. So I wanted to know from the audience, are there questions?
I have a question regarding the data analysis, mainly on the microbiome, because it's a problem that I'm having. How much you did shotgun microbiome analysis, or in the soil microbiome?
Yeah.
You did the whole sequence or the ITS sequence, or how did you evaluate the microbiome diversity?
Well, I think I may be wrong, but I think it's mostly what we're doing is 16S sequencing, okay, and also mycorrhizal AMF sequencing.
Okay, so you're doing bacteria.
Yeah.
Okay, and okay, how many samples do you have for?
Okay, so we have. I didn't do the sampling, but we have 5 field sites and 1 control condition site. We have 6 genotypes, and each genotype is repeated 3 times. So we have 3 reps, and in each rep, we take the samples from 4 plants, which are bulked. And these are shipped to Ireland. Most of them are in Ireland already. shipping the samples from Turkey is a nightmare because not because of the borders, and we have to decide if there is money to do a second year of sampling, or if that's actually what was committed in the project was this first year of sampling.
No, because we are, I will explain later, but because we have more, I mean, higher sample size, but we have problems for the analysis, recruiting people to do the analysis. So it was my big project problem, so that's.
The analysis will be handled mostly by the staff at UCD in Ireland, with help from Katowice and also from Zaragoza. Yeah, please.
I think one thing that was very interesting, because there's a lot of similarities with the wheat project, is to have a joint meeting.
That would be so nice. We had a meeting in Dublin.
Yeah, you had a meeting in Dublin when we were in Italy. But it would be nice, but also I wonder if there is the option, if to share some field sites or location, like for example, you grow in Dublin.
Yep.
So we grow in Dublin. And if it is in a similar field location, because Tancredi is doing microbiome in yours, he's doing is he doing it? No, Manila and Maghrib's doing the microbiome, but he's doing the data analysis. We're using exactly the same protocol. Everything is the same. It's very standardized, so it could be very nice to have some trials, at least one, maybe more, that are in a common region.
You mean for microbiome?
For wheat and bird, but you're finished your microbiome, is that it this year?
Sorry?
You've completed all the microbiome?
In principle, yes. So we don't need more samples because, I mean, we have, Tancredi is going to analyze quite a good number of samples, but he still has to decide whether he has enough budget for the second year, or if he fails for whatever reason, then we look at the sample again. So we are planting the barley dedicated plots for microbiome this year again, just in case.
I think you have more, well, we went to 5 sites, but 2 failed this year. But you have more sites, but then we have more genotypes. I think we have maybe 12 common genotypes per site.
Mm-hmm.
I just think it would be nice. And also as a follow-on later, you mentioned the association mapping populations, you know.
Well, yeah, that is something that we're constructing. We have been discussing whether to go with the double haploid method to create these populations. We did the process last year, but not this year. But it's something that if it is done, it will be ready for right after the project. That would be for the next project.
Yeah, but it could be right. And another project, which is for the confusion, I think she saw another project we have online called Wheat Health. And what we're doing with John and is we're screening the NAM parents for disease, but also for microbiome.
Okay.
And if we find that we don't know yet, that analysis is ongoing, Tancredi's involved in it. If we find differences, then we're going to look at the population, the relationship between the microbiome and plant health in the population. It'd be very nice to look at that environment.
Well, definitely. I mean, you have Sonia and Tancredi really close, so please communicate with them. And I think we will be quite open to have
meeting. Would be
we are going to have the annual meeting in Dundee, actually, in February.
Okay.
That far from it.
Yeah, no, so maybe I could come to that.
We will have the Barley-Genome Net small meeting, and then 2 projects. Best crop and our project will have meetings together, back to back.
Thanks.
But yeah, thanks, because there are many things in common, as you said. Yes.
You mentioned that some long races have more yield in some environments than the more modern barley that have cultivated now.
This class, productive class. Yeah.
But this is in low nitrogen.
Low nitrogen.
Yes,
I showed Spain's results because Turkey results were not so encouraging in Turkey, the land races did quite bad. But on the other hand, the old heritage varieties of Central European barley did quite well. And what I didn't show is introgressional lines that we have of very exotic in the lead material barley. These are doing really well.
But very well across low nitrogen conditions or compared to potentially high nitrogen?
In good conditions.
So you have
introgression lines.
So you have some of the heritage strains that do in low nitrogen as well as the normal ones in high nitrogen?
In Turkey, the best ones were local Czechs and modern varieties, and there were some old heritage varieties which did almost as good as the local Czechs and the modern varieties. But this is just 2 locations. We haven't analyzed yet Ireland and Finland. In Fiorenzuola, everything was wiped out by the floodings, as happened to the wheat probably. So we have 4 locations so far, and we need a second year. I mean, I wouldn't jump to conclusions from any of these results, really.
You mentioned also, can I ask another question?
Yeah. That's
you just mentioned that you have stomatal conductance, not a number, stomatal density that vary between land masses on your panels, in your panel, if I well understood. I think it's very interesting because in mice, some colleagues of us showed that on genetic progress experiment, meaning that you look for commercial hybrids that have been across the year, they showed that there is some progress, general progress for yield and so on, tolerance to stress, but there is not too much progress. There is some ecophysiological target like stomatal conductance for which there is no there is variability but no progress at all, meaning that there is a window for breeding for these squats.
Yeah, I agree. It's something we don't know much about, probably because we didn't have, you do research where you can put the light on. We didn't have good tools to study stomata. So now we have, I think, the Lycor 600 that Diego was showing one picture, and we have these microscopes that you can actually study the density of stomata. And as you said, you find really nice genotypic differences. And we have to relate that with water use efficiency, drought tolerance, but there is a lot to do in that respect. I agree.
Probably I'm wrong, but I think I remember that in a conference someone said that the stomata density did not correlate with the aperture, with the conductance. So saying that, but I remember that from that maybe having more stomata didn't mean that they were going to be more.
Of course, probably there is the genetics of stomatal density has nothing to do with response to APA or things like that. But what you heard probably was right and applied to that crop and that material.
Yeah, maybe.
That's
Yeah,
the same as for our experiments. So we really need to expand the knowledge on the field implications of stomatal density and stomatal behavior, which may be completely independent. Thank you.
So I would like to thank all speakers and guardians, of course. So now we have a little break, and it will be at 4:10. We go back to the general room for the last session. Thank you. 4:10. So 25 minutes.